Hi, welcome to exercise 14, the sense organs. Well, sometimes I call it special senses, sometimes I just call it eyes and ears because that's all we'll be talking about. At any rate, use this PowerPoint as you read through exercise 14, which does start on page 143. And be sure to turn up your volume if needed. Well, you probably didn't hear that if you needed to turn your volume up. Oh well. Exercise 14 does start on page 143, although that's just the instructional objectives that I still wasn't going through. On page 144, there is an introduction, and they talk about how the sense organs are those that allow us to see, hear, taste, touch, and smell. And those are generally referred to as our special senses, although realistically in this lab, we're going to concentrate on vision and hearing, or eyes and ears. The lab book does go into some methods, and they're going to make it sound like we're actually going to, oh my god, dissect an eyeball, which we used to do, and it's really kind of fun, but we're just going to be looking at models at this point. So if you'll flip over to figure 14.2, we'll start identifying structures there. We'll come back and talk about some of the rest of the terms over the course of the lab exercise. Page 144 does have figure 14.1, which does have some structures of the eyes as well, although that one gets into a little too much anatomy detail. So we're going to need to know the structures on figure 14.2. Wait a minute, figure 14.2 on page 146 isn't labeled. Yeah, I know, I'm sorry, I was just being evil at that point. But officially we do need to know the structures on figure 14.2 rather than 14.1. So I'll give you a few minutes to label them, and then we'll be going through the definitions and showing them on some of our models that we do have in lab. The eyeball does have three general layers to it. The first layer is going to be the sclera, which is the white part of the eye. It's also the outermost part of the eye. But when you're looking at somebody's eyeball and you do see the white of the eye, that would be the sclera. That does extend all the way around to the back side of the eye. No demonstrations, please, but we all know somebody who can roll their eyes into the back of their head and all you see is the white part. All you see is the sclera. Technically, the cornea is a modified section of the sclera. It's modified to be clear and transparent and would take up most of the front part of the eye. It's probably best that the cornea is modified to be clear and transparent, because if you had white of the eye all the way across the front, you wouldn't be able to see anything. So we do have to have the clear, transparent front portion of the eye so that light can go inside. The cornea is where you'd put your contact lens on your eyeball. Do be careful when you remove your contacts that you don't scratch the cornea and other damaging things. The second layer of the eye is the choroid layer. It's the middle layer, and it does have a lot of blood vessels. On our model in lab, on the right side of the picture, you can see the red arteries and the blue veins. I haven't decided why they removed that off of the left side, although you can kind of make out some of the lumps and bumps where those arteries and veins would be, but that choroid layer should have all the blood vessels on it. Some other structures that you can see from this point of view of the eyeball is the iris. The iris is the colored portion of the eye. On this model, it happens to be a blue eye, but yours might be brown or green or hazel or gray or pink if you're albino. Honestly, our model does have 1/4 of the iris removed on kind of the right-hand side of it so that you can see the underlying deeper blood vessels and the choroid layer. Still not sure why they took that off, but what the heck, they took the iris off so that you can see the underlying structures. The pupil is the hole in the iris, and it is an opening that lets in more or less light. On my picture, you can see a little bit of a reflection off of the lens, but usually the pupil is the black spot in the middle of the eyeball. Now, honestly, it's really not a black structure so much as a hole in the eyeball. The inside of the eye doesn't have any light in it, so when you look into the depths of the eye, it looks black. It's just dark in there. I still think we need some Christmas lights at the back of our eyeballs to light them up so they'd be blue or pink or green. Oh, well, maybe not. But the reason it looks like a black structure is because there's no light on the inside of the eyeball, so it just looks like a black structure if you're looking in the mirror, but really it's a hole that would let in more or less light. If you get in a situation where you've got a lot of light, the pupil will constrict so that you let in less light. On the other hand, if you turn the lights out, oh my gosh, it got dark, the pupil will open and let in more light. The pupil would dilate and let in more or less light. On this picture, I've removed another top chunk of the eyeball there. Well, dissected our model? Okay, I just removed the lid off of it. Most of what you see in the front of the eye is the aqueous humor. That's the space in the front part of the eye. There is actually some gel-like goo in the aqueous humor. The vitreous humor is the gel-like goo in the back of the eye. Most of what you thought of as the inside of the eyeball is probably vitreous humor. Honestly, I kind of remember it as aqueous humor. A is at the front of the alphabet. A is at the front of the eyeball. The vitreous humor, V, is at the back of the alphabet, and the vitreous humor would be at the back of the eyeball. Dissecting a little further and removing the vitreous humor, we finally get to the third layer of the eye. That would be the retina layer, and it's the innermost layer that does have the rods and cones. We'll talk more about the rods and the cones soon, but the rods are going to do black and white vision, and they need relatively little light. The cones would do color vision, and they need generally a lot of light. Ultimately, those photoreceptors from lecture, the rods and the cones send a message to the brain, and your brain can figure out what exactly you're seeing. On the front side of the eyeball, the lens is a clear marble-like structure. It's not really marble, but in real life it is a clear transparent portion again, and it will change shape to focus light on the retina layer. Light would come in through the front of the eye, the lens would change shape and focus the light rays on the retina layer. How the lens focuses is a bit of science major stuff. The lens can be fatter, and you would use a fatter, thicker lens to look at things that are up close. If you need to look at something that's farther away, there are ciliary bodies and suspensory ligaments that will pull on the edges of the lens and make it a little thinner or flatter. Figure 14.3 is trying to show some of that, and you can kind of make out the A part does have the thicker, fatter lens, whereas the B portion has the more elongated, flatter lens where you'd be looking at something farther away. Honestly, it's a bit of science major magic. For non-science majors, we'll leave it as the lens changes shape to focus light on the retina without exactly talking about how specifically the science major magic and physics and angles of refraction actually work. The lab book just points out the ciliary bodies, but in real life and lecture class, there were some suspensory ligaments. Together, they work to pull on the lens to change its shape. On the figure here, on the model, you can see the little white lines. Those are actually supposed to be the suspensory ligaments that are attached to the ciliary body that would pull out to the sides and flatten out the shape of the lens. If the ciliary body and the suspensory ligaments aren't pulling, well, then there's not as much tension, and the lens can be a little fatter so that you would see things up close. Yeah, well, non-science major stuff. We pull on the lens to change its shape, and we're not really going to mention exactly how it does that. I said earlier the retina layer was the innermost layer, and it did have the rods and the cones. Those rods and cones do send messages along the optic nerve, which takes light messages from the rods and the cones to the brain. Now, for most of the eye that you can see here, the kind of snakeskin pattern and kind of red color, that would be the retina layer with its rods and its cones, and they'd be sending the information over to the optic nerve. In one small little area, there's what they call a blind spot. That's where the optic nerve leaves the eyeball itself. There's no rods, there's no cones, there's just optic nerve. We call that the blind spot. Wait a minute, Miss Snaples. I don't see any spot where I can't see anything. Well, honestly, the blind spot in your right eye is covered for by your left eye, and vice versa. Wait a minute, Miss Snaples. I've closed one eye and I still don't see a spot where I'm blind. Well, honestly, your brain is very good about merging information from the sides. We'll see a blind spot test coming up shortly, and hopefully you can prove to yourself that you do in fact have a blind spot, but your brain is very good about covering up for it. Technically, there is a spot back there where the optic nerve leaves. There's only optic nerve, there's no rods, there's no cones, so there's no vision. But your brain's used to that and covers for it quite easily. The last thing on figure 14.2 is the fovea, or fovea centralis. It's an area with a high number or high concentration of cones. The lab book says there's so many cones that there are no rods. I'm not going to guarantee that one or two of the rods didn't slip in there somewhere, but for the most part the fovea just has the cones which do the color vision. So it would actually be an area where you've got very good vision. It's a little hard to tell in the picture because I had to turn it sideways to actually show the fovea back there, but light rays do come into the front of the eye, go through the cornea, through the aqueous humor, through the pupil, through the lens, through the vitreous humor, and land right smack on the fovea. And if you're looking at something straight ahead of you, it generally does land straight on the fovea. If you're looking at something off to the side in your peripheral vision, you're still going to have some rods and some cones, and you'll still be able to see whatever the object is. But if you're really going to look at something, you do put it straight in front of your face, and the light rays land on the fovea, so you do have a field of good vision. One of the things I don't like about this model, really the fovea is not an area on the back of the vitreous humor, which is certainly what it looks like on our model. Really, the fovea is an area of the retina that has a high number of cones, and our model's not quite anatomically correct. Yeah, well, we'll just have to go with it, but keep in mind that that fovea with the high number of cones really should be on the retina and not the vitreous humor. I have mentioned it already, but on page 145 the lab book does talk about the innermost layer being the retina, which is actually some nervous tissue and an extension of the optic nerve. It does have a whole bunch of layers which do have the rods and the cones. The rods are, well, rod-shaped, just like their name says, and ultimately they do black and white vision. They need relatively little light. If you turn off the light, or if you wake up early in the morning and you don't have the light on, you walk in your closet and try to pull out a shirt, who knows what color you'll end up with, because it's a low light situation and you'd be using your rods which do more of the black and white vision. You might be able to tell if your shirt is a light colored shirt or a dark colored shirt, but there's no telling if it's going to be blue or green, because your cones are what does color vision. C is for cookie. Oh, well, on Sesame Street, C may be for cookie, but in biology for non-science majors, C is for cone and C is for color vision. Cones do color vision. In lecture, we talked about there being 3 different types of cones: red cones, blue cones, and green cones, and all of the colors that you see are a combination of those red, blue, and green cones. And they would do the color vision. They would send the messages to the brain on the optic nerve, aside from that little blind spot in there. And don't forget the fovea is the area that does have a lot of cones, so you see really well right there. At the bottom of page 145, number 5, the experiment continues on page 147. This time, look at the distant object. Look at the far away, look at the far away, look at the far away. Close your eyes. When you open your eyes, do you see your finger, or do you see far away? It kind of varies. Different people will see different things. I would guess that if you're looking at your finger, you will probably, when you open your eyes, you will probably see your finger. If you're looking at the far object, and you close your eyes, you probably should see the far object because that's kind of where your lens is set. Although, some people will report the opposite there. The way the anatomy works is when you close your eyes, you might start relaxing some of the muscles. When you relax the ciliary body and the suspensory ligaments, that actually expands out, which pulls on the lens. It's kind of one of those science major magic things. But ultimately, when your eyes are relaxed, that should flatten out the lens, and a lot of people will see the far object no matter which one they were looking at. And what I've seen from students in class is it does vary. About half the class will see their finger all the time. About half the class will, well, I guess a third of the class will see their finger all the time. A third of the class will see the far object all the time. And a third of the class will see whatever they were looking at. So it does get a little interesting. Part number 3 says in moderate light, one student should look at an object at least 20 feet away, and another student should examine the subject's pupils. That's the one where you might need a little hand mirror there. Ideally, you would look at something across the room, look at something far away. You grab a little hand mirror, you look at your pupils. Generally, if you're looking at something far away, your pupils are probably constricted and small. On the other hand, when you get that hand mirror out and you look at it really close in front of your face, that's going to be like looking at something close in front of your face. And your pupils will generally dilate just a little, very quickly, and that's what they're trying to get you to do, is note the size of the pupils. When you look at something far away, should be constricted. When you look at something close up, it should be slightly dilated. Has the function of the mammalian eye. And they say as 2 students begin the following parts of this lab exercise, honestly, we don't need 2 students. And really, even in my on-campus lab, I don't recommend that you have 2 students because we'll be staring in each other's eyes. And yeah, that sounds like sexual harassment again. So you can actually do these on your own. Most of it you can do by yourself anyway. And for one part, we might need a little hand mirror so we can watch some of the movements in your eyes. But the first part says that accommodation, the eye is capable of focusing on near and far objects. The focus is possible through accommodation, which is where that ciliary body and suspensory ligaments are changing the shape of the lens to make it fatter or flatter. The lens can stretch and relax because of ligament and muscle control. So to observe and practice, what you should do is face an object approximately 20 feet away. Look at something across the room. Hold your hand with outspread fingers at a distance of about half a foot from your face. Really doesn't matter if you're 6 inches or an entire foot, stick your finger in front of your face. What you're supposed to do is look at your fingers, look at your finger, look at your finger, close your eyes. When you open your eyes, do you see your finger or do you see the distant object? On page 147, number 5 continues on with B, astigmatism. This is the name for a condition in which the refracting surface of the cornea or lens is not a perfect sphere, but is curved irregularly. It might have some lumps and or bumps on it. Honestly, the way you check for an astigmatism is using an astigmatism chart. Like seen here in the picture, each of the 3 lines is going to be the same density, darkness, intensity, blackness. If you're seeing any of the lines that are looking a little darker or a little lighter, that might be a sign of an astigmatism. Ideally, look at it straight on. The lab book says at a distance of 20 feet, but honestly they're lying about that one. As long as you're looking at the astigmatism chart straight on, it should work. Ideally, cover one eye and see. Oh my gosh, oh wait, the 11 and the 5 are popping out for me. The 9 and the 3 just went a little lighter. Yeah, I do have a little problem with an astigmatism. Honestly, if you go to the eye doctor's office and they start doing the thing where they're asking you, is A darker than B? Flip, flip, flip. Is B darker than A? Flip, flip, flip. Which one's darker, A or B? they're checking for an astigmatism. And when they're flipping through the little gadgets, they are sometimes making A darker, sometimes making B darker, and I think sometimes they're making them equal intensity and just trying to fake you out. Some days I guess pretty well, and they say I don't have an astigmatism. Some days I guess pretty badly, and they say I do have a little itty-bitty teeny tiny, don't bother to correct it, astigmatism. I have run these astigmatism charts in lab for a good many years. One of the semesters with some anatomy students, one of the girls looked at it. Now she knew she had a problem. She had glasses. They were to correct her astigmatism, but when she looked at the astigmatism chart, she couldn't see the lines at 3 and 9. She didn't realize how bad of an astigmatism she had. They had flat out disappeared on her. That was pretty freaky there. If you don't have an astigmatism, the lab book says that you can place a small beaker or glass of water in front of your eye and repeat the experiment. The refraction of the light through the water and the glass will make some of the lines pop out a little better than others. Just be careful, don't spill the water on your computer, obviously. Number 5, letter C, is a blind spot test. You'll probably want to use the figure in the lab book rather than looking at this one. I can't get this one to do quite what it's supposed to. But with the left eye closed, what you do is you hold the lab book about 20 inches from your face and look at the cross. Ideally, you want it lined up so that your nose would kind of hit in between the cross and the circle. They should be kind of even, and on my picture here, the dot is a little too far down, I think, to make it work quite right. But to try to find your blind spot, close the left eye, or hold it closed, whichever. Hold the lab book about 20 inches in front of your face. Look at the cross. As you move the lab book closer to your face, stare at the cross. Out of your peripheral vision, oh my gosh, did the circle just disappear? At about 12 to 18 inches, the circle will actually disappear on me. I can't see it anymore. It's really kind of freaky there. But eventually, you get it so that the circle is in the blind spot of your right eye, and your brain merges the white of the rest of the screen over that blind spot and compensates for it, and you just see a white spot instead of the black spot. There's also a link at the bottom to test and find your blind spot in each eye. They've got a few more things to try, and they've got some different color schemes and different patterns and things like that, which is kind of cool. If you'd like, you can also try covering your right eye and staring at the dot and seeing if you can make the cross disappear. Now the cross is a little bit bigger, so it will be harder to make that cross disappear. Hint, if you're trying it at home, turn the lab book upside down. Close your right eye. Stare at the cross, and with the lab book upside down, you can make the circle disappear a lot easier than making the cross disappear. Try it. If you can't make it work, it is kind of fussy on the angle. You do want it straight in front of your face, a little too far up and you're not going to see the blind spot, a little too far down and you won't see it, too far to the right, too far to the left. If you bring it in too fast, you might miss your blind spot. If you really can't find it, you still have one. I'm sorry. You do have one. Whether or not you can prove it to yourself may be a different question. Page 147 continues on with number 5, letter D, demonstration of the Snellen eye chart. There is actually an eye chart in your book, although I haven't figured out exactly where you need to stand for that. I'll try and update that in the future. Ultimately, there is a link down here at the bottom of this page. You're welcome to click on that one. They do have instructions for print out the eye chart, how to measure it in millimeters, do some conversion, and figure out how many feet back you should stand for your newfound eye chart. Most of the ones in real life in the doctor's office have been set up so that you would stand at 20 feet. I'll be calling out the numbers based on the 20 feet, but the eye chart in your lab book is not quite to the right scale for 20 feet, so don't use that one to diagnose yourself. You're going to come up nearsighted at that point. But what they've got is a little chart here, and ideally you would stand at the right distance, cover one eye, and read the lowest line you can. This does not work real well sitting in front of the computer screen. You can probably read the lowest line, line number 11. Well, maybe line number 10, I don't know. It's not coming in really clear, but you should be able to print out a real eye chart, stand at 20 feet, and read the lowest line you can. Ideally, you would be reading line 8, which is 20/20. 20/20 we'll talk about in lecture, but the first number is always going to be 20. That's because you stand at 20 feet. The second number refers to where other people can read that line. So 20/20 means you're standing at 20 feet and other people are standing at 20 feet and reading what you can read. I really don't have exactly 20/20 vision. Without my glasses, I'm probably closer to line 5 and 20/40, which means I can stand at 20 feet and read line 5, whereas most people can be a little farther back at 40 feet and still read it. I would be closer to the eye chart. I would be nearsighted. Nearsighted really does mean you can see things up close. It just kills me when somebody says they're nearsighted and they can see things that are far away. Did you think about what you were saying? If you're nearsighted, you are going to see things up close, not far away. That would be farsighted. Farsighted, they're not showing on this picture here, but lines 9, 10, and 11 would all be farsighted. Line 9 is actually supposed to be about 20/15. My brother happens to be very farsighted, and he can read line 9 with no problems. He'd be at 20 feet, whereas other people would have to be at 15 feet. He'd be farther back. He'd be farsighted. He can see far away. Honestly, I think he can read line 11, which would be about 25. He can stand at 20 feet and read what most people have to be at 5 feet, really close to the eye chart to see. So he's actually severely farsighted. There's really not a spot in the lab book to record that, but a lot of people are kind of interested in their vision, and many people do have some problems, so that's at least what the doctor was looking at. Again, click the link, follow the directions, and you too can find out your vision and visual acuity.
Okay, it took a little research and actually having my lab book and finding a ruler, but I did measure the letter E. It was 60 millimeters high. You multiply by some number, divide by something else, and that gives you the meters, the length in meters to stand from your lab book. Oh, wait, like any American's going to need to know that number. Working some more math, converting things out. If you'll stand at 160 inches, that's 13 feet 10 inches from your lab book, you can use the chart, the eye chart on page 147 to actually check your vision. You would still read it in 20/20 or 20/40 or 20/10 units, but because our eye chart is a little smaller than the standard one at the doctor's office, you would need to stand at 13 feet 10 inches. That ideally would be the measurement between your eyeballs and the chart rather than your toes in the chart. Yeah, I wouldn't spaz over it that much. Stand about 13, maybe 14 feet and you should be fine.
The lab book does go on to the next special sense would be hearing and ears. On page 150 there is figure 14.6, which is nicely labeled, but it's got a few extra things that we really don't need to know about, like an internal carotid artery and a styloid bone and some other stuff. So we're going to be learning figure 14.7. The unlabeled figure 14.7. Yeah, just call me evil again. The only trick is figure 14.7 does not have a cochlea. You will need to add the cochlea. It is fair game on a lab quiz or lab practical. I'll give you a few minutes to write down the names of the structures, and then we'll be talking about them. Keep in mind, it wasn't real easy to put all of the words that match up with the lines, so have fun trying to match things up. The numbers really do refer to the lines, and a few of my on-campus students have some trouble matching up the lines, so I tried to put more of the blue and the red lines that would match up with the original lines on the picture. Really, the problem is on the right side. The first line is really far up, and I think people miss the first line with the semicircular canals. So be sure you're really starting on the first line rather than the second. Figure 14.7 in the lab book really is the figure of a right ear. Yeah, I know it's on the left side of the page, but it really is a right ear. Unfortunately, our model in lab is a left ear, so things will look a little backwards. Don't panic, but we're still going with my backwards ear here. Starting on the left side of figure 14.7, there is the pinna. The pinna is the outer ear. It is made up of a lot of elastic cartilage, and so you can flip it and you can flick it, and it'll just pop back into place anytime you start playing with it there. If you do pierce your ears, you would pierce into the cartilage. Oh, owie. Yeah, well, you'd actually be piercing the pinna, or the outer ear. Another structure you're probably familiar with, although you might not have called it that, is the auditory canal. It is the opening in the ear that does lead to the inner ear. That's the one that all the doctors and official people will tell you not to put anything in your auditory canal that's smaller than your elbow. Wait a minute, I can't get my elbow in the auditory canal? Yeah, that's actually their point. They tell you not to stick anything in the auditory canal, because if you do jab things farther than my little green arrow goes, you could hit the next structure in, which is the tympanic membrane, and you could actually damage your eardrum. Yeah, I know it wasn't Johnson & Johnson, the makers of Q-tips, who said that not to put anything smaller than your elbow in your ear, because, yeah, they've got Q-tips. On the other hand, they do tell you to be careful with them, because you don't want to burst out your eardrum or damage it in any way. Here's a close-up of the tympanic membrane, otherwise known as by its non-science major term as the eardrum. Really, in real life, the tympanic membrane is a membrane, and it does vibrate, kind of like the head of a drum, ear drum. Yeah, well, the sound vibrations would come in and vibrate that eardrum or that tympanic membrane. In plastic model life, it's a piece of plastic, and it doesn't do very much. But in real life, it should actually vibrate and transfer the sound vibrations to the next structure. The next structure would be some ear bones. There are 3 ear bones that are kind of connected between the tympanic membrane themselves and the cochlea. The 3 ear bones are the malleus, the incus, and the stapes. You might have heard of these by their high school names: the hammer, the anvil, and the stirrup. Unfortunately, we're in college now, and their college names are the malleus, the incus, and the stapes. The malleus is what we used to call the hammer. The anvil, hammer hit the anvil, the anvil is now the incus. The stirrup, hey, it does kind of look like a little itty-bitty teeny tiny stirrup. The stirrup is now the stapes. The 3 ear bones really just kind of run together in the ear, and honestly, I just run them together as one word: the malleus, incus, and stapes. I just kind of act like it's all one piece there. But the first ear bone would be the malleus, the second would be the incus, and the third would be the stapes. They're all going to take the vibrations from the eardrum, from the tympanic membrane, and transfer the vibrations to the cochlea. The cochlea is a fluid-filled shell that has hairs that bend in response to sound waves. You can see the remains of the stapes, stirrup, coming in on the bottom of the picture, and it is transferring the sound waves to the cochlea, the fluid-filled shell. In lecture, we talk about exactly which hairs sense which pitch The close hairs do a soprano pitch, and the farther hairs, more in the coiled part of the cochlea, would pick up the bass or the longer waves, the low-pitched. So the cochlea does have the hairs that bend in response to sound waves. Also in the same general area are 3 semicircular canals. The 3 semicircular canals have hairs that bend in response to movement. One of the semicircular canals will deal with movement in a left and right plane. If you move left and right, you can imagine the little A semicircular canal going off and hairs sloshing back and forth. If you go forward and back, I don't know, maybe it's the semicircular canal with the B on it that takes care of the back and forth movement. The other semicircular canal would do an up and down movement. If you're riding in an elevator, that would set off the 3rd semicircular canal, hairs that bend in response to movement. The last structure on figure 14.7 is the vestibularcochlear nerve, which takes equilibrium and hearing information to the brain. If you can read through the Latin of the name, you might get some hints of it. Really, in lecture we talked about the 3 semicircular canals coming together in the vestibule. It's just where the 3 semicircular canals come together and about where that stapes is attached. The vestibule takes information from the semicircular canals, which were dealing with equilibrium. The cochlea was that fluid-filled shell that did hearing. So if you can read through the Latin, the vestibular part really refers to the equilibrium parts, and the cochlea really refers to the hearing parts, and that yellow thing on the model there would be the vestibularcochlear nerve that takes equilibrium and hearing information to the brain. There were 2 other items that were omitted off of the figure. One of them, that stapes there, or stirrup, is hooked to a window. Technically, the stapes has a oval shape to it, and it's hooked to an oval window. The 3rd line on the right-hand side of the picture was actually an oval window. The 5th line on the right-hand side of the picture, oh my god, is that a little circle? Yeah, there's a round window underneath. I'm going to assume you can tell round from oval, from triangle, from square, and we're just going to omit those. On page 151, there are some questions. Number 1 said a clouded lens is a cataract and results from the breakdown of protein in the lens in old age, overexposure to bright light, trauma, diabetes, and so forth. It does result from a lot of different things, although straining your eyes, studying for lab practicals, no, not yet, but soon, studying for lab practicals does not actually cause cataracts. Cataracts really are a cloudy lens. In order to treat cataracts, what portion of the eye must be removed? Well, the part that's not working quite right? The lens, perhaps? Yeah, you'd remove the lens to fix cataracts. The trick is, if your lens is clouded, you're not getting enough light going through, so not everything is focusing on the retina layer, so eventually if the lens clouded over entirely and no light got through, you wouldn't be able to see anything. You'd, in effect, go blind. So, yeah, you'd probably want to remove that cloudy lens. So, number 2, how could a person who had a cataract operation be able to focus an image on the retina? If you've taken out the lens, what you want to do is you want to replace it with a synthetic lens. It is a little more difficult than plug and play and changing the batteries in your mouse or laptop. It's a little more difficult than that. They can't just take out an entire lens and put in a fresh brand spanking new one. Honestly, they cut out and hollow out most of your existing lens, and they fill the shell with some synthetic stuff that hardens into a lens, but it all stays in the suspensory ligaments because you do leave a little chunk of your original lens there. Number 3, the closest distance at which an object appears in sharp focus is called the near point or near point of accommodation. They don't really say what you should do here, but if you'll take a pen or a pencil, where you can read number 2 pencil or big pen, stick that in front of your face. See how close you can get the pen or pencil to your nose and still be able to read the writing. At that point, try to guesstimate how close the pen or pencil is to your eyeball. That would be your near point of accommodation. And generally, at age, say, 20 years old, the near point of accommodation is about 3.5 inches. You can get the pen pretty close to your eye before you go cross-eyed and can't really see anything. At 30 years of age, however, it's about 4.5 inches. You're pulling back just a little bit. At 40 years of age, the normal near point of accommodation is getting even further back, 6.75. I've done this lab exercise for some time. I can feel my near point of accommodation going further and further back. I'm closer to 45 now, and it's closer to about 8 inches out. Used to be closer to the 3 or 4 inches. Yes, it's a form of age discrimination. For that matter, how can one account for these changes? Age. The older you get, the less the lens wants to change shape, and so you pull things further and further back to see them. What's your near point of accommodation? You'll have to guesstimate the distance between the pen, pencil, and your eyeball there. Does your near point fall within the figures listed above? Hopefully. Again, don't diagnose yourself based on some non-science major data, but if you are worried about things, do make an appointment with your eye doctor and ask him about it. Number 4, list the sequence of structures through which sound waves and nerve impulses pass from the moment a word is spoken until you actually perceive that word. We're omitting that. Just cross that one off. Although, really, the sound waves have to go through the pinna, through the external auditory canal, bang on the tympanic membrane, vibrate the malleus, vibrate the incus, vibrate the stapes, vibrate the cochlea, slosh the little hairs, then you've got to have some nerve impulses going through the vestibularcochlear nerve to the brain. Yeah, there's a reason we were omitting that question. It's entirely too long for a multiple choice question. But if you do think about it, you probably can answer it. Number 5. Humans have become accustomed to movement in the horizontal plane, which stimulates certain semicircular canals, but we're unused to vertical movements parallel to the long axis of the body. Explain why some people will become nauseous and vomit from the rocking of a boat, elevator ride, or fast ride on a merry-go-round. That's because we're unused to vertical movements parallel to the long axis of the body. Circle, star, highlight something. Write down about that we're unused to vertical movements. We do have 3 semicircular canals. One of them handles left and right movements, which we do a lot of. One of them handles forward and back motion, which we do a lot of. One of them handles up and down. We don't use that one quite as much. If something's going to go wrong, it's going to be the one we don't use quite as much, parallel to the long axis of the body with the vertical movements, the up and down thing. Will generally spaz. If somebody's going to spaz, it'd be the up and down motion. Let's face it, if you're going to spaz over the left and right, you're probably not going to get very far in life. You're probably going to be barfing the whole time. So yeah, that would be a really bad one, and I don't know if you survived that one. Yeah, that's what we're talking about. That's actually all we've got for the special senses. Thanks for listening.